working in the field of optoacoustic imaging since 2010. He is a senior scientist and group leader at the Institute for Biomedical Engineering and the Institute of Pharmacology and Toxicology at the University of Zurich and ETH. Previously, he did a postdoc at the Institute of Biological and Biomedical Imaging in Munich at the Helmholtz Centrum and contributed to the development of new optoacoustic systems and processing algorithms, as well as to the demonstration of new biomedical applications in cancer, cardiovascular biology, and neuroscience. He has co-authored more than 70 papers on this topic so far. And he is joined by Paul Wrede, who is currently at the Max Planck and ETH Center for Learning Systems in Stuttgart as a PhD student. Paul received his bachelor's degree in biomedical engineering from the Technical University in Chemnitz and his master's in biomedical technologies from the Eberhard Karls University in Tübingen. He worked at the EFW in Dresden from 2017 to 2019 and did research on synthetic chemical propelled microrobots as well as sperm bots. So after 1 year research stay at the ETH, he joined the Max Planck and ETH Center for Learning Systems and now aims to design microscale robots that revolutionize the treatment and diagnosis of diseases. Besides the next generation of smart microrobots, he is especially interested in visualizing these tiny man-made machines in vivo. So consequently, his research interests include microrobotics, bioimaging, micro and nanotechnologies, as well as personalized medicine. And we will start off the presentation with Louis, and we're looking forward to your talk.
As I was saying, thank you very much for the introduction. So we split the presentation into two parts. So mainly we will introduce the technology, what is optoacoustic imaging. Maybe some of you know it already, some of you maybe the first time that you hear about it. And particularly in the aspect of how to image dynamics and what we can do to image dynamics with it. And then we will show some applications in super resolution imaging and in also in tracking of microrobots, which is the part that Paul will present. So I will start with the first part, which will take 25 minutes approximately, and then Paul will take over. Okay, so first of all, I would like to start with the motivation, with the general motivation of why using optoacoustics. So many of you probably know about the advantages of optical imaging, so particularly optical microscopy. You just were introducing something about super resolution microscopy. And basically, it's a very useful tool in molecular biology because we have protein-specific contrast, because we have dynamics in the cells, and we can see a lot of information with fluorescence contrast or with other types of optical contrast. The problem is that we can only use this information, or we can only resolve things with optics when we are in the very shallow regions. And the reason of that is that as we go deep into tissues, light starts to scatter, so it starts to be diffused, and everything becomes blurry. So an analogy to this is what happens, for example, what you see here in the left-hand side of this slide, what happens when you have fog in the air. So if you don't have fog and you're seeing a picture, for example, the lights of the streets and of cars in a city, so you can resolve very nicely everything, but as long as you have fog, so everything becomes blurry. You can see that there is some light, but you cannot resolve it. You cannot distinguish anything, and this is what we need with imaging, right? So, and this is the same in vivo. So this is a summary here of the different optical imaging techniques in a paper from 2010. So basically, we have very high resolution when we have very shallow depths. So this penetration depth typically where 10 transport mean free path is around 1 millimeter. And then, no, sorry, the mean free path is around this one is around 1 millimeter. So everything, as you go to deeper than 1 millimeter, starts to get, we start very fast to lose the resolution. And then this is not, I mean, it's not okay. So the idea is to push things in this regime here with optoacoustics as much as we can. So take down the resolution when we go deep into tissues, because it's not that light doesn't reach deep, it's light gets diffused when it goes deep, but light's still there, so we can interrogate tissues with light at depths of even centimeters. So how does optoacoustic work? So the very basic principle was discovered or invented by Alexander Graham Bell like 1 century ago, or even more than that. And the idea is that when we shine light, when we shine a pulse light into something, and this gets absorbed, this gets thermalized, it gets expanded, and then it generates an ultrasound wave. So this is, I can repeat again the animation. So we have a signal like this. So it's like an ultrasound signal, it's a time resolved signal, it's pressure as a function of time. And the signal arrives at the given time, which corresponds to the time of flight between the absorber, in this case, and the transducer that we use to collect the signal. So this is the information that we have. And then once we use this information, for example, collecting signals all around the sample, and we use some reconstruction algorithms, so we can see, for example, an entire mouse with optical contrast. So this is what you see here is a cross-section of the mouse. And what you mainly see here are the blood vessels in the mouse, which is where we have the contrast with optoacoustics. What mainly generates contrast in tissues is blood. Now we can exploit the advantages of optical contrast, as I mentioned, to extract a lot of information. And we like to say that optoacoustics is like a 5D imaging modality, because we have a 3-dimensional imaging. We have also the time dimension. And apart from that, we can do imaging at different wavelengths. So we can change the wavelength of the laser, and then we can exploit this spectral dimension, for example, to resolve what kind of components are in a tissue. And this is what we see here. We can resolve for the distribution of oxyhemoglobin in red, deoxy in blue, and we can even see like a melanin spot here, like in skin pigmentation, which is plotted in yellow here. And this is because every component has a different absorption spectrum that we can distinguish based on the wavelength information. So we have 3 spatial dimensions. We have the temporal dimension, and I'll focus on that in this presentation. And we also have the wavelength dimension, or the spectral dimension. So we have 5D, 5-dimensional imaging. Okay, the other important thing of optoacoustics is that it's a multi-scale imaging modality. So as I mentioned, if we do optical microscopy, if we do optical imaging, this is mainly used as a microscopy imaging modality. So we can see something like very high resolution, but very shallow. In the depths typically less than 1 millimeter. Even 2 photon or the optical microscope that can reach deeper, they cannot really reach deeper than 1 millimeter. In optoacoustics, we can use the same approach as is used in optical imaging, but instead of getting a fluorescence response, for example, we get an optoacoustic response. But in the end, we can focus light, and we can get the resolution with light. And this regime here is what's called optical resolution optoacoustic imaging, because basically it's not based on ultrasound. Ultrasound is only to collect the signal and to collect the information. We also have, of course, some depth information with ultrasound, but basically these 2D images formed with optical resolution. So we focus the beam and we scan it around, and then we have the same resolution or similar as we get with optics. So we can use optoacoustic in this regime, in the same regime as optical microscopy, but with a different type of contrast. But we can also go deeper, and as we go deeper, then we have to exploit the ultrasound beam forming, the ultrasound reconstruction to get an image. So because at those depths, light, as I mentioned, light gets diffused. But ultrasound doesn't scatter so much, so then we can reconstruct with ultrasound. And these are 2 examples. So one is the microvasculature in the skin for depths of 2-3 millimeters. This range is what's called mesoscopy, because it's between microscopy and macroscopy, like full body tomographic imaging. And the image that you have in the right is an image of the carotid artery in humans that is 1 centimeter even deeper than that. And we can still resolve it with optoacoustics, so we can reach these depths and get this images at these depths. So in this range, as I mentioned, also the resolution is given by acoustic beam forming. So it's acoustic resolution is what's called acoustic resolution optoacoustic imaging. And as a rule of thumb, we have the resolution that we can achieve with different imaging systems. There are actually many different types of approaches. Is typically 1 divided by 200 the imaging depth. This is a rule of thumb. So as we go deep, we have attenuation increases with frequency, and then we have to use lower frequencies, and then we lose the resolution. Okay, so the good thing of this multi-scale imaging property is that we can kind of bridge the gap between different modalities. Optical imaging is mainly working in the microscopy regime, and then we have other imaging modalities like full body imaging modalities, X-ray, MRI, that work don't do microscopy, but they can provide full body imaging, even of humans, but if of humans or of mice or other animals. And with optoacoustics, we can go in between. So we can go from the microscopy regime to this macroscopy regime with the same type of contrast, which is very important. So we can interrogate tissues with the same type of contrast at different spatial scales. And this is not the full picture. So we will have a look also at the temporal resolution, what is represented here as a volumetric imaging rate. Optoacoustics is also very competitive. So we can go also very fast, and we can actually even outperform most of the imaging modalities that are existing there. And this is the part where I'm going to focus in the presentation, particularly in the dynamic imaging capability of optoacoustics and the applications of that. Okay, so why can we go that fast? I mean, we can go that fast because, as I mentioned, we can use a laser to excite the tissue. We can generate ultrasound, and then we have to detect signals. So in the first systems, the way to go was use a single element transducer, like detect one signal and then scan it around. But one thing that we can do is to use what's called an array of transducers, which can collect many signals simultaneously. So this, for example, this that you see in the picture is the typical arrays that we use that have 512 elements. This means that we can collect 512 images with only 512 signals with only one laser pulse. And these 512 signals are sufficient to make an image. So in the end, we can get an imaging rate that is given by the pulse repetition rate of the laser. The typical time resolution that we can get is 100 Hertz, and we can get very nice information here. For example, we can image the beating heart of the mouse, which is actually very fast. We're talking about 7 beats per second. And we can then use we can also scan around, of course, losing temporal resolution and see the full body image of the mouse. And then we can have this multi-scale temporal resolution. We can go, if we focus in one small region, we can image very fast. And if we scan, we lose temporal resolution, but we can increase the field of view. So we have this also what we call multi-scale dynamic imaging capability. Okay, so as I mentioned, typically the laser repetition frequencies that we use are 100 Hertz. So this is a limitation of the pulse repetition frequency of the laser regarding the maximum temporal resolution that you can achieve. But it's not only that, then it's we can actually use higher pulse repetition frequencies of the laser. But then, of course, we have a lot of data to acquire, and basically the electronics or the electronics, typical acquisition electronics cannot handle that. So to be able to image at higher frame rates, what we did, instead of acquiring all these signals that I mentioned, 512 signals with each laser pulse. So what we did is to do a sparse acquisition of the elements of the array, so that we acquire only a set of elements for every laser pulse. So what you see here visualized in different colors. So basically, with the first laser pulse, we would acquire the blue channels, the blue signals, with the second, with the green ones, the third one, the red ones, and so on. And by doing it like this, we can handle the data, we can handle higher imaging rates, and we can also, of course, we have to then change the reconstruction algorithms. So we do this model-based inversion with advanced regularization methods, but we can, in the end, get an image at higher, even at kilohertz frame rates. And I'm going to show two examples. One is a freely imaging zebrafish, which was done by my colleague Ali Esbeck. And you want to see it like this. The video is very much slowed down, so the whole video is like half a second or less than that. And I mean, now you see very smooth movement of the fish, but this, if you see it in real time, actually with your eye, it's impossible to distinguish the movement. So this is actually very fast motion, freely swimming zebrafish larva. So this is, I think, a nice example, and this is another example, which is a work led by my colleague Charles Zoy. And this is visualizing the Langendorf heart, which is the isolated heart, also at this very high temporal resolution. And what we could see is that we have these spiral waves, like this, what's called scroll waves, which are typical of arrhythmias. So here we induce these arrhythmias in the heart, and we could see the result, which is this scroll waves. So this electromechanical wave propagation is also something very difficult to visualize with other modality, because we need these imaging rates. We need to go to the kilohertz imaging rate, and here we could see it. So this also is a very important application of this high frame rate imaging that we just published last year in PNAS. Okay, so Good. So as I mentioned, so what is absorbing, what is generating contrast in optoacoustics? So typically, as I mentioned, what we see is blood. So hemoglobin is very highly absorbing, and then it generates a lot of contrast. So whatever is absorbing light is generating ultrasound, and this is how optoacoustics works basically. So if we have something very absorbing, we will have a strong contrast, a bright spot in the image. And this is a good thing or a bad thing, because we have endogenous contrast, so we can see the vasculature without injecting anything. There are also, as you see here, other endogenous chromophores like melanin, lipids, and water. But also, it's not so good if we want to see some exogenous contrast, because it's we have a background and then we lose a bit the sensitivity, right? But there are actually many different types of contrast agents. So we have molecules, nanoparticles, genetic reporters, fluorescent proteins, or chromoproteins, for example. And generally, anything absorbing light we can use as a contrast agent, exogenously administered into the animal. Okay, so I'm going to show you some examples how to use this dynamics to see like this contrast enhanced optoacoustic imaging. So one is to see the perfusion of ICG in a tumor. So this thing that you see here, so this is a three-dimensional image of a tumor. And what you see here is like the three views. And now I'm going to show the video basically showing when we inject ICG. So you see the ICG coming, and then you will see that it slowly starts accumulating in this region, which arguably means that we have like this enhanced permeability and retention effect in the tumor. We can also exploit the optoacoustic capability of using the multispectral optoacoustic imaging, this spectral dimension, to also resolve oxygenation in the tumor and see hypoxia in the tumor. So this is, I think, a very powerful tool for cancer research. And another example that I show here, I like this image, and I show it always in the presentation. So this is injection. Let's see how the contrast agent perfuses in the heart of the mouse. So we see the image of the heart in real time. And then we inject the ICG, in this case, again, ICG, and then we will see that it goes first to the right ventricle. And then sometime after, you will see a signal increase in the left ventricle. So this is, we can see how long it takes for the blood, or for the contrast agent, for that matter, to go through the lung circuit, so the pulmonary circulation. And this is very useful as well, very useful information, because we have these dynamics on something very fast, that is a beating heart of the mouse. And with this, we can extract the parameter, for example, that is called the pulmonary transit time. And we saw also that this parameter it changes in different disease models. And this is also some kind of information that is difficult to get other ways. Now, the thing that I want to draw your attention to is that when we use a contrast agent, like in this case, I can play the video again. What you see is that basically we see a continuous. So we see that we don't see individual objects there, we see something continuous. And the reason is that typically the contrast agents in optoacoustics are these molecules, small nanoparticles. So we're not seeing individual objects, let's say. So we're seeing many of them, and then what we see a continuous signal. And this is a characteristic of optoacoustics, basically, is different from ultrasound, because ultrasound, we have this discontinuous image, we have this speckle image. And in optoacoustics, we wouldn't have this unless we have a very sparse distribution of source, what I call source or absorbers here. So, for example, if we have an image like this, and we do an optoacoustic image, so we have a lot of optoacoustic sources. Optoacoustic sources can be molecules, can be anything. So we see something continuous. If we see something, we have a sparse distribution of sources, we would see something discontinuous, but I will show that this is actually very challenging, and this is basically what we're trying to achieve. And the other thing that you see in this slide is that when we have basically, in this case, it's using a transducer array that has only 90-degree angular coverage. So the image that you obtain is something like this, which doesn't look like the original image. And this is something that is intrinsic of any tomographic imaging modality, is called the limited view problem. So basically, when we don't have enough information around the object, we cannot reconstruct it properly, because we're not covering all the spatial frequencies that are there, all the directions. But if we have it, let's assume that instead of having a continuous distribution of sources, or many sources that we can assume that is a continuous thing, we have a sparse distribution of sources. You see that the shape of the object looks very much like the original one. And this is something that we can also exploit if we manage to find the right sources or the right things that we can detect individually in optoacoustic image. And I'll come back to this, but I just wanted to introduce this problem first with this example. Okay, so the problem is that, as I mentioned, we have endogenous contrast, and if we want to compare, for example, this is like a graph showing the different type of contrast agents of optoacoustics, and what we see here is the molar extinction coefficient or the absorption per unit. Per unit means per molecule or per nanoparticles. So what you see in the left is the molecules, in the right is the nanoparticles. So we see the natural absorbers here in this graph, which are the red blood cells. So basically, we see that red blood cells are absorbed more than all the other types of contrast agents that we use in optoacoustics. And red blood cells are very dense in tissue. So this is a video showing how basically red blood cells, and you see that red blood cells basically are 50% approximately of blood. So the concentration of red blood cells is very high. That's why we see this continuous absorption or continuous signal in optoacoustic images. So typically, in a voxel of in a typical voxel of an optoacoustic image, talking about 100 microns, we would have 10,000. We can have up to 10,000 red blood cells, which is a lot. But we cannot distinguish them individually, at least when we go deep into optoacoustics, into tissues. So is there any chance that we can see individual things with optoacoustics? Well, this is what we're trying, and we will see that there are interesting applications. Okay, so one thing that we have done is to remove the blood to avoid this problem of the blood background. So for this, we use an intracardiac perfusion model where this is a trans vivo or ex vivo experiment. So basically, we substitute the blood to inject perfusion through the heart, what's called ACSF, artificial cerebrospinal fluid. And this is something physiological that keeps the mouse, at least physiologically, the heart continues beating for some time. So we can still inject something and see it in the mouse, but without the blood. So you see that the signal, this is the original signal, and this is the signal when we remove the blood. There's still some blood accumulating in some regions that we cannot remove, but the background signal is reduced a lot. And in these circumstances, what we did is to inject B16 melanocytes, which are circulating tumor cells, and then is where we start seeing them individually. So these individual dots that you see here are actually individual cells. So we can actually use optoacoustics to track and visualize in real time the distribution of something individual. So this was the first time that we achieved something like this. The problem is that these cells are very large. So it's very interesting to use this, for example, to study metastasis, because this is what happens in metastasis. We have these circulating tumor cells that flow through the vasculature and then get stuck somewhere, and then this region is where a secondary tumor is developed. But of course, if we want to use this approach to do something in vivo, something that flows through the vasculature to do some therapy or to do some enhanced images, as I will show. So this is not an option because this will get arrested in the capillaries very easily. Now, one thing that we can do with this approach is to do, well, I have it's 20 minutes now. I think I still have 5 minutes. It's fine. So is to exploit this to do super resolution imaging. Again, we have to find the right particles. These cells are nice because we can see them individually. The problem is that they get stuck in the capillaries. But we could do super resolution imaging if we find something similar, but that flows continuously through the vessels without being arrested. So I'm going to show you the concept. I think you know it because actually you have a talk next week. So the idea of super resolution is that when we have an imaging system, and we try to quantify the resolution, basically what we think is the full width half maximum of given by diffraction, right? But if we think, for example, of something very wide, a very big full width half maximum, like a big mountain, wide mountain, okay, this doesn't mean that we cannot accurately determine, for example, the position of the peak of the mountain. The position of the peak of the mountain is very accurately measured, even if the width of the point spread function of the system is very wide, right? And this is the concept of super resolution, which was suggested for optical microscopy. So you have individual fluorophores that are activated on and off, and then we see individual dots in the signals, in the images, in a sequence of images. So then we can localize, we can detect the position of these individual dots, and we can build an image based on the localized points. So if we have enough points, you can think that if we have a few points, we have something that doesn't look like an image, but as we add more and more points, in the end, it will look like a continuous image. And we can have like an image that has in the end the resolution that is given by the accuracy in the localization of these points. So this is the idea of localization imaging. You probably know it because this is used in microscopy. So the idea is to use the same in optoacoustics. So if we use, if we have something that we can detect individually, and this is moving through the vasculature, then in the end, we can localize a lot of dots, and we can build an image like this. This is a phantom. So this is very easy because here we can use very big particles. Now it's doing it in vivo is not, it's a bit more challenging. But you see that we can resolve an image with this approach, and if we compare it with the standard optoacoustic image, which is what you see in the right, we see that the localization image, that is this one in the left, it gives you much higher resolution. And on the other hand, you see that also we can see the shape of the knot much nicer than in the optoacoustic image. And the reason of this is what I mentioned before about the limited view. So when we have like a continuous absorption, like we have in standard optoacoustic image, because of this tomographic problem, this limited view tomographic problem, there are some parts that we're missing, unless we, of course, cover the whole thing, but this, in practical cases, sometimes is not possible, right? But with this approach, with localization, we can actually resolve also not only high resolution, but also more accurately the shape of the object. Okay, so after some time, we finally managed to find the particles. And what we did, basically, is to use these dichlorometane microdroplets, which are liquid microdroplets, are relatively small, so are 5 microns on average. So they're sufficiently small to flow through the vasculature. We didn't see any capillary arrest with these droplets. And we labeled them, or we didn't label them, we included in the DCM, basically this dye, this IR780 dye, that has like a very good extinction coefficient, absorbs much more than blood. And also it was possible to to dissolve it in the chlorometane with a very, very high concentration. Where, as a comparison, so the concentration of the dye in the droplet is like 200 millimolar. The concentration of hemoglobin in red blood cells is 2 millimolar. And the extinction coefficient is also much higher than hemoglobin for 780, which is the peak absorption of the dye. So we have microdroplets, something that is 5 microns, that has a dye that is super concentrated and super absorbing. So if this doesn't work, then nothing's going to work. But luckily it worked, and then we could inject them these droplets in vivo, intravenously, and you will see now that we will see them individually. So we see them flowing. And this is when I saw this image the first time, I was actually super, super happy, because it took us a lot of time to find this. But just seeing the droplets, actually, you can do many things with this. That's why I was so happy. So one of the things that we can do is basically to use this approach that I mentioned to reconstruct an image. So we can put together all the dots that we localize, and then we can see an image like this, which, as you see, so it has much more details than the original optoacoustic image. So this is a comparison. So basically, the standard optoacoustic image is here in the left. The localization, what we call localization optoacoustic tomography image, is in the right. So we see the microvasculature much nicely. This is a section of the like a coronal section of the brain. So we see different layers in the cortex. And we estimated that the resolution is typically what we can achieve is in the order of 15, 20 microns, like 17, 22 that you see here. And this is deep, like 3 millimeters deep approximately into the tissue, which is something that we cannot reach with, for example, with optical microscopy. Okay, but there's more than that, because we can use this information, this flow of droplets, not only to reconstruct an image with super resolution methods, but also we can use that to measure the blood flow, also in 3 dimensions. And we can get an image like this, which is a map of the blood flow. So we see here a slow velocity plotted in blue, larger velocity in red, like it's in the major arteries. And basically, so we can resolve, or we can quantify all this velocity distribution even at 40 millimeters per second, which is very fast. So yeah. So this is additional information. We can not only resolve better the image, we can quantify the blood flow, and also we can exploit the multispectral capability of optoacoustics and multi-wavelength, this spectroscopic mixing, also to resolve oxygenation in the microvasculature. And this is an image. This is a model of stroke that was done in collaboration with Mohamed Elanki here from Zurich. And you see that not only we see lower distribution of vessels in the stroke affected area, but also we see less oxygenation, which is also very useful information. So this is summarizing this example, basically. Again, this is the model of stroke that we image. So we can see in both cases, on the one hand, that if we compare the left, which is the region affected by stroke, and the right, that is a healthy region, let's say. So we have much less vascular density. We also have lower velocity, and we also have lower oxygenation, and we have significant differences in all cases. Okay, so this is the end of my part. I think I took 3 minutes more than what I should, but I hope that I have convinced you that optoacoustics can provide very powerful imaging capabilities. So if you have never heard about optoacoustics, maybe I will convince you to investigate a bit more about the technique. And particularly, one important thing is that we can provide very high temporal resolution of entire volumes. And this is what I mainly focus in this presentation. So it was possible, it was feasible to individually detect micron-sized objects. And this, I showed the example of cells and also microparticles, and then Paul will show the example of microrobots. And this opens new possibilities. So if we develop new types of contrast agents, so this can enhance the capabilities of optoacoustics a lot. So in this case, I showed the example of super resolution imaging, but it's not only that. We can also do blood flow velocity mapping. We can also do oxygenation of the microvasculature and many other things, believe me. It's the future is bright.
So yeah, after Luis also already now introduced you to optoacoustics, I will start introducing you now to the world of microrobotics and how optoacoustics can facilitate the applications of microrobotics for medical applications. First of all, I would like to maybe show you a bit what is a microrobot, because a microrobot is very different from a normal robot as we would imagine it. So it doesn't have arms or legs or looks like an animal like these robots you sometimes see on Facebook. So they look very different because they have to be much smaller. So when we talk about microrobots, we talk about size scales ranging from below 1 millimeter and being a bit bigger than 1 micron. And my aim mainly is to develop these machines for medical applications. And here I showed you like something that we envision as a community to do at some point. So we would like to have an imaging system and an actuation system. In the best case, they would even be integrated into each other so that we only would have one system that can do both. Then we would have the physician standing next to the patient, simply injecting our microrobots. Here, for example, I put an example of a vascular application where we inject the microrobots into the vessels of the patient. And then we would, under biomedical imaging, guide our microrobots towards the diseased area. In this case, it could be, for example, a plug inside the vessel that you want to dissolve. And then we use our microrobots to targetedly deliver a drug or physically dissolve the blood clot and thereby do minimal damage to the vessel and decrease the needed drug concentration by a lot, which would then in turn also decreases possible side effects. So when we design such microrobots, we kind of do a bottom-up approach. So we want to first see a medical condition that we want to target. And going out from that, we then start designing our microrobots, because then we know what is the disease area, so which body area we are working on, what are the fluid conditions there, flows, and so on. And then we start integrating medical functions into our microrobots. How can we actuate them? The best possible actuation method for the given environments. Also, what's the best possible imaging modality we can use so that we can integrate some contrast agents to our microrobots, think about possible retrieval options such as biodegradability or targeted movement back to the injection site. And of course, also one major thing is the biocompatibility of our microrobots, so that we don't cause any harm to the body. After all these steps and in vitro validation, we would then go to an in vivo model where we test all these capabilities. And then in the end, this is what we envision at the moment, we would start with clinical trials. So at the moment, the community is in this stage of the in vivo validation stage. So there are more and more papers coming up where people start to validate their developed microrobots in small rodents. And here, one major issue is the medical imaging, because as I already told, these microrobots are very, very small. So we talk about micron scale, and we need some precise imaging of them with high imaging frame rates, also high imaging speed, so high temporal resolution, because you want to know the position of these microrobots at any time so that we can really precisely navigate them through the body. So for that, optoacoustic imaging comes in very handy, as Louis already explained, because it shows very high resolution paired with very high frame rates. So one of the first group to show optoacoustic imaging for microrobots was a group led by Liang Wang and Wei Gao in Caltech. And they showed these micromotors, which had a size of around 100 micrometer. And they were coated. It was a magnesium particle coated with different layers, here gold, a drug, and perilene. And then they encapsulated several of these micromotors into a bigger capsule, which was then swallowed by the mouse. And by this way, they got into the stomach of the mouse, where using NIR radiation, they could dissolve the capsule around these microrobots and free them. And then using a chemical reaction with the gastric acid, they were able to dissolve this perilene layer and also free the drug. And so that it gets released inside the intestines. And then these microrobots propel through the intestines using a chemical reaction. And you can see they're tracking here over time course of 7.5 hours through the whole intestines using optoacoustic imaging. Also, the group could show that they could see their microrobots until a depth of around 40 to 50 millimeter, which is quite deep. Next, there was also an approach with so-called biohybrid microrobots. So this is a different type of microrobots where we kind of use biological materials. This could be sperms, bacteria, or cells. And we decorate them with different particles, for example, to be able to control them. In this case, they decorated spirulina platensia with iron oxide nanoparticles, and then also added a dopamine layer as an optoacoustic contrast agent. Also, this bacteria type has a size of around 100 micrometer. And due to the spiral shape, using this iron oxide nanoparticles, they could use a Helmholtz-Kohl setup to apply a rotating magnetic field, which would then actuate these robots towards the desired area. They then injected these spirulina platensis microrobots under the skin of a mouse and used a rotating magnetic field to propel them towards target site. And then these bacteria there caused a bacterial infection, which they treated using an NIR laser light, which was absorbed by the iron oxide nanoparticles to cause a local heating, which then killed the bacteria and therefore also the bacterial infection. Other approaches involve much more complex geometries where we use 3D printed microrobots here as a cell carrier. And in this case, this group used this carrier to embed cells into it and carry stem cells towards different places inside the vena cava of a mouse, also using optoacoustic imaging, but also ultrasound imaging. Here also the actuation method was magnetic, and the size of these microrobots was also around 100 micrometer. Another group from Dresden also showed another type of microrobots, which is particle-based again, but this time we call them Janus particles, which means, so they reassemble the Roman god Janus, which has two faces. So also here in this case, these particles kind of have two faces because we have a silicon oxide surface on one side, and on the other side, we have a metal surface. In this case, this was nickel and gold. The gold was used to increase the biocompatibility of the microrobots and serve as an optoacoustic contrast agent, while the nickel was used with them for magnetic actuation again, where we have these big magnetic coil setups. They then injected these robots into the uterus and bladder of the mouse and were able to move them under optoacoustic and ultrasound imaging inside the bladder and the uterus. So now if we think back to the first application I showed, I told that we are kind of aiming also for the vasculature, because from there we can really reach almost any destination inside the body. So we are very free to move. But the recent robots I showed were quite large, so they would never fit inside the vasculature. They were all around 100 micrometer. And if we think about the picture or the video also, Luis showed these red blood cells are very, very small. So they can really squeeze through all the narrow vessels we have in our body. And their size is around 5 to 8 micron. So this is kind of the size scale we are aiming for. But this, of course, also makes the imaging much, much harder using optoacoustic imaging, because the hemoglobin gives very high signal, which is good because then we can see the vessels. So we really know where we are moving. But of course, our microrobots should give even more signal than that. So this is kind of the thing we had in mind when we started our project. And we started with 5, 10, and 20 micrometer Janus particles. So we also decorated them with nickel and gold on one side to increase biocompatibility and allow for magnetic guidance with the nickel. And also the gold should be used as optoacoustic contrast agent, while the other side of our microrobots were decorated with lipo-ICG. Lipo-ICG is short for liposome ICG, which means that we have a liposome, and in the lipid layer of this liposome, we have ICG encapsulated. As Luis already showed, ICG is a very well-known contrast agent also for optoacoustics. We then measured the absorption of our microrobots, first without ICG, where we get this relatively flat graph here, and then also with ICG, where we get this nice absorption peak here around 800 nanometer wavelength light, which showed us that we really functionalized our microrobots with this lipo-ICG. Also, the very cool thing about this particle-based approach is that we can fabricate these microrobots with very high yield and very reproducible, which is really needed for medical applications, as we have to inject a lot of these microrobots to get significant drug loads and a useful drug load into the patient. So next, we started validating our microrobots for the real optoacoustic imaging. For that, we embedded our microrobots into agarrows and placed them onto our transducer array, where we received then the acoustic signal, and we shined laser light from the top. And here we used pulse repetition rate for the laser of 10 Hertz. So also our imaging rate is 10 frames per second. We then measured the CNR of our microrobots, and in only agarrows, we could see that they gave a relatively high CNR, and also as expected, the bigger the microrobots were, the higher was their CNR. To validate that we are really seeing single microrobots, we also took some reference microscope images, as you can see here on the bottom, and then compared them to our optoacoustic images. And the reason why these spots here look much bigger in the optoacoustic image than compared to the microscope image is simply the resolution of our system, which is around 120 micrometer, which is why these spots here appear as 120 micrometer size, since these microrobots are far below the resolution limit. So then, because we're also aiming for applications in the vasculature, we also added blood into the system here, and then calculated the CNR again. And as expected, the CNR dropped drastically. But we were still able to distinguish especially the 10 and 20 micrometer microrobots. But what we could see is that the difference between the lipo-ICG and non-lipo-ICG coated microrobots almost diminished. But we still kept going with the lipo-ICG coated microrobots, as for further projects, lipo-ICG is very handy because we can also load drugs to the liposomes, which is then the next step for applications. So we then tested our microrobots in more realistic environments. So we here extracted the coronary vessel of an ex vivo pig heart and placed it on our detection array. Then we perfused the whole heart, the vessels with pig blood using a peristaltic pump and injected our microrobots into the blood, and then imaged the microrobots, as you can see here in this video. So as you see, we could really detect the 5, 10, and 20 micrometer microrobots. So you see all these small dots flowing through the vessels. But still, we were not very satisfied with the CNR we got. So we decided to do some post-processing. And here we decided to go with singular visual decomposition filtering, short SVD. And here we take the initial images we took, and then extracted the static vessel background from these images by subtracting it. And then we were left with the dynamically changing, over time dynamically changing microrobot signal. And then we overlaid this extracted microrobot signal again onto a static vessel image, which we color coded differently. So we had 2 different color codings. And then we end up with these nice images. So to show that also in a video, you see it here. So now it's very easy to detect the microrobots. And in this case, we also placed a magnet on the end of the vessel to slow our microrobots down, which then already indicates the ability to magnetically manipulate these particles. With these results, we now went into the animal experiment. And for that, we first took a localizer image of our mice. So we positioned it on our transducer cup, and we wanted to see the circle of Willis structure here, which is a very prominent vascular structure inside the brain. And then we injected the microrobots into the heart of the mouse. So we opened the rib cage and then directly injected into the heart so that the still pumping heart could pump our microrobots into the brain. And then for the 5 micrometer microrobots, you could here nicely see them flow through the brain. And this we also repeated for 10 micrometer microrobots, where you also see them moving through the brain here in these green dots. And also the 20 micrometer microrobots we were able to detect. So after that, we then also wanted to demonstrate the capability to really manipulate these microrobots, because that's like the key part of our microrobotic systems, that we can really move them around using different types of actuation mechanisms. And here we applied magnetic actuation. So we simply placed a permanent bar magnet on the brain of the mouse, and then pulled these microrobots towards the bar magnet. And this we also did for all the different sizes we tested. So 5, 10, and of course also the 20 micrometer microrobots. With that, I would like to conclude my part of this talk and summarize the main novelties of this work we did. So we were able to really scale down the size of our microrobots up to 20 times smaller than what was previously shown with optoacoustic imaging for microrobots. And this enabled us to really use our microrobots in the microvasculature, which now opens up many new possibilities for the microrobotic field, so that we can now really work on meaningful medical applications and functions. And we were also able to show that we are able to do 3D imaging of these microrobots with high frame rates of 10 frames per second, which is really important for precise navigation. Yeah, with that, I would like to end my talk and acknowledge everyone who worked on the project, especially Professor Metin Siti and Professor Daniel Rosansky, who really made this whole work possible, and also, of course, my close collaborators, for example, Luis, who was also speaking today. Thank you.
Thank you very much, Paul, for showing these applications with the microrobots. That's really interesting, and I'm really curious to see how this will go on and develop, and what applications will emerge from that for medical therapies. Do we have any questions from the audience for Paul? Johanna, I can see you have a question.
Yeah, thanks, Paul. This is a very interesting, fascinating work. I was wondering what happens with those microrobots after you have applied them, and sort of what's the conceptual idea there? And you think you can use these imaging technologies that you've developed here to sort of track the whole lifetime of these microrobots in the animal?
Yeah, exactly. So the thing is really that now that we solved the imaging issue, we are currently working on very precise actuation mechanisms, also acoustically based now, not only magnetics, because as you saw, these magnetic setups are very big. So we try to scale that down using acoustic actuation. And then the idea for these specific particles that we used here would be to move them, or to collect them and move them back towards the injection site, or move them somewhere where we can easily retrieve them. because these micro robots are not biodegradable at the moment. But there are some approaches showing also biodegradable robots, which are, for example, made of liposomes or other lipids. And these could then even be left inside the body and would degrade over time. This could even be a mechanism for releasing drugs, for example, over time.
Great, thank you.
Yes, yeah, you were exactly talking about a question I had here. What is the preciseness currently of the actuation and then moving the particles to a certain site?
So that's highly dependent on the actuation mechanism we are using. So as you already saw, also in the previous papers I showed, there are like some main modalities. I would say the most used one is magnetic actuation, where we can be really precise. So we can really position the microrobots. I would say we have a micrometer precision there. But these setups are very bulky, so you need these big Helmholtz coils to apply different magnetic fields. You can modulate the magnetic fields then also. And other options are chemical actuation, as shown in this first paper, where they use the microrobots inside the stomach. This is quite unprecise, as they kind of randomly move. Only way to control them would be a chemoattractant, so that they have some sort pH value, for example, where they go towards to, which can also be used for tumors, for example, as we have different pHs there. But the other one is acoustic. This is something that is starting to develop now, so it's relatively new, acoustic actuation. And this is something, we are currently looking into, and there we are in the very beginning. So we have resolutions for the preciseness of actuation around 250 microns there.
Thanks a lot, Paul. Are there more questions from the audience? If so, please raise your hand or unmute yourself. So while people think, I have a question actually that is not related to the imaging directly, but since I have not been following this field, what is the current regulatory situation around these micro robots? Are there any micro robots already in clinical applications or FDA approved? And if yes, for what application?
So no, microrobots is still a very young field. So I think we are now really the first in vivo trials going on now. But I know some companies start to include this approach into their R&D also. So I guess maybe in the next 5 years we will maybe see the first really applications in the humans, or the first trials at least. But there's still a way to go, because as you already mentioned, like we now have kind of the imaging, but we now have to really combine this with the actuation and with the safety considerations, and also for the regulatory process that makes it even more complicated, because there's no such product yet. So it is likely that there are new regulations needed for these devices, as they are not passive devices but active, but on a very different scale. So it's not yet super clear what are the exact regulations here.